Hello guys, welcome to Ninja Explains.m. My name is Mubarak and I am your tutor.
Physiology Lecture 1. Homeostasis. A controlled system consisting of a collection of interconnected cells that function to maintain a physical or chemical property of the internal environment relatively constant. Homeostasis. Temperature. Electrolyte balance. pH. Fluid, etc. Stability of internal environmental variable is achieved by balancing inputs and outputs. It depends not upon the absolute magnitudes of opposing inputs and outputs but only upon the balance between them. Mechanisms of homeostasis. Two main mechanisms are involved. 1. Negative feedback mechanism, PSH direction, opposite the original change, thus lead to stability. Therefore the common homeostatic mechanisms in the body. E.g. temperature regulation. Positive feedback mechanism, sets off a trail of events which increases the disturbance even further. Thus, positive feedback does not favor stability but often, abruptly displaces a system away from its steady state of operating point. Positive feedback may sometimes be beneficial, e.g. as occurs in coagulation, childbirth and depolarization. Generalizations of homeostatic control systems. Stability of an internal environmental, balancing inputs and outputs. It's not the absolute magnitudes of the inputs and outputs. In negative feedback systems, push the variable in a direction opposite the original change. NO complete constancy, narrow range of normal values depending on the external environmental conditions. The more precise the homeostatic mechanism for controlling a variable, the narrower will be the range of normal values. Thus an ideal, complete constancy, can never be achieved. It is not possible for everything to be maintained relatively constant, there is therefore a hierarchy of importance. For example, body temperature, changes in skin blood flow and skeletal muscle contractions. Acidity of blood, changes in CO2 of blood. Acclimatization. Acclimatization is the ability of an individual to adapt to a particular environmental stress in an enhanced manner without any change in genetic endowment but by prolonged exposure to the stress. In other words, it can be viewed as an environmentally induced enhancement of an already existing genetically based homeostasis. Repeated or prolonged exposure to the environmental stress, up to some maximum limit, enhances the responsiveness of the system to that particular type of stress. Some acclimatization, for instance heat acclimatization, is completely reversible since if daily exposures are discontinued within a relatively short time, sweating rate during future tests would reverse to pre-acclimatized value. However, if the acclimatization is induced very early in the individual's life during the critical period of development of the structure of response, it may be irreversible. E.g. barrel-shaped chest of the natives of Andes Mountain does not represent a genetic difference between them and their low-land relatives, it remains even after the individual moves to low-land environment later in life and stays there. This developmental acclimatization changes the form not only of anatomical changes as in the example above, but physiological responses as well. Total body water and fluid compartments. Within various tissues and organs in a whole animal are found membrane-separated fluid compartments. Collectively these are known as total body water, TBW. There is considerable variation in total body water between species, ages, sexes, amount of adipose tissue, nutritional states and other conditions. Lean adult non-herbivore, about 70% of its body weight. Water content is highest in the newborn animal, declines rapidly at first, and then declines slowly. The total water content in a fat animal will be lower than a lean one due to exceptionally low water content in fat tissues, only 40%. Within the TBW are major solute cations, Na+, K+, H+, Ca+, and Mg++, and anions, Cl-, HCO3-, PO43. solute distributend between various subdivisions of body water by active and passive processes. Generally, there are two types of body water mainly separated by cell membrane, including extracellular and intercellular fluid. ECF/internal environment. Life is presumed to originate from the primordial ocean/sea. The ocean provided both a mean of nourishment and transporting toxic waste from organismal activity away from them. Multicellular organisms evolved to enclose within them fluids which serve the same function as the sea, helping the organism in transiting from land to sea. The SEA, constituting body fluid outside the cell, forms the internal environment of the organism. Every cell is surrounded by fluid, the characteristics and composition of which must be regulated with a narrow range. ECF 20% of BW and 33.3% TBW. Subdivisions of ECF plasma 5% BW and 8% TBW. Interstitial, tissue, fluid, ISF, outside the blood vessels and bathe the cells. Also include lymph 8% BW and 12.5% TBW. Fluids of dense connective tissue as in skin, ligaments, tendons, and bones. Same as interstitial fluid, but water exchange is so slow, behaving as a separate compartment. 10% TBW. Transcellular fluid, separated from other compartment B epithelial membranes, include synovial fluid, CSF, ocular fluid, pleural fluids, peritoneal fluids, pericardial fluid, and fluids in hollow organs, digestive, urogenital, and respiratory tracts. 1% BW, 2.5% TBW. TCF are different from other fluid because they are produced by secretory processes. Intracellular fluid found in living cells 66.6% TBW, 40% BW. Differs in composition even in different regions of one cell because of many organelles present in a cell. It however considered a single compartment. Composition of body fluids generally, subdivisions of ECF, except transcellular fluid, are nearly identical in composition of electrolytes and small molecules, and differ mainly in protein. Na+, HCO3, and Cl are the chief electrolyte of the ECF. The plasma is rich in protein, but the passes with difficulty into the ISF. ICF, K+ and Mg+ principal cations, PO4 and protein as major anions. Electrolytes are the main solutes in the body fluids for maintenance of acid-base balance. They maintain proper osmolality and volume of body fluids. The concentration of certain electrolytes determine their specific physiologic function, for example Ca++ and neuromuscular excitability. Exchange of fluids and electrolytes between compartments O2, nutrients and H2O must be transported into cells while waste metabolites are transported out. The differences in composition of body fluid is largely due to the compartments separating them. ICF separated from ISF by cell membrane. Plasma by capillary wall. Forces producing movement of substances between compartment. Forces producing the movement of molecules across barriers are either active or passive. Passive forces cause movement through pressure or concentration differences or gradient, and do not require expenditure of energy by the cell. Active transport is where the cell provides energy or specific molecule to move a substance across the membrane, active and vesicular transport. Passive transport mechanisms. Bulk flow, solvent drag, due to pressure or concentration different both solute and solvent are moved through a membrane, as occurs capillaries. Filtration, due to pressure or energy difference on two sides of a membrane, acting as a sieve. Molecules forced through the membrane based on size. This also occur at the capillary end. Diffusion, molecules are in random motion and the speed of movement depends on temp. These movement causes collision between moles. A greater concentration in an area in solution or on one side of the membrane tend to lead greater collision per unit area, distributing the mole rapidly in the solvent. Distribution slows as solute concentration tend to equalize. When passage is equal in all directions, equilibrium is said to have been attained. Influx equals efflux, i.e. net influx equals zero. Diffusion occurs rapidly when smaller particles are involved. Simple and facilitated diffusion are found. Facilitated diffusion. If the passage of a molecule across a membrane requires a carrier mole, no energy is required. Concentration gradient is still relevant here. Glucose is transported into cells partly by this process. Simple diffusion is expressed by Fick's law. The magnitude of diffusion tendency from one region to the other, J, is directly proportional to the cross-sectional area across which diffusion is taking place, A, the concentration or chemical gradient, C equals C1, C2, and inversely proportional to the thickness of the diffusion boundary or distance to be traveled, J equals dAc/X. Factors determining diffusion of molecules across biological membrane. Lipid solubility, N2, O2, CO2, alcohol, steroid hormones and weak organic acids. Water solubility, for example water. When water-soluble moles are too large to go through the channels, they are facilitated by carrier proteins. Electrical charge. Size of mole. Selective permeability of membrane. Osmosis. A type of diffusion, but allows the movement of only solvent or water through a membrane that restricts the passage of solute while allowing solvent. The total solute concentration of a solution is known as its osmolality/osmolarity of a solution. The difference in osmolality of solutions on either side of a membrane gives an idea of the magnitude of force, osmotic force, that will cause water movement across a membrane. Thus the pressure necessary to prevent solvent migration is the osmotic pressure of a solution. Normal osmolality of body fluids approximate 290 milliosmoles per kilogram, osm/kg. Cells behave like osmotic system, allowing solvents and preventing entry of solutes. If cells are placed in solutions with osmolality similar to theirs, water movement will be at equal rate. The cell is said to be an isotonic/isosmotic solution. If placed in solution whose osmolality is less than that of the cell, net influx of water will occur into the cell. The cell is said to be placed in hypotonic/hyposmotic solution. If the solution outside the cell is more concentrated, it is said to be in hypertonic/hyperosmotic solution. Plasma is isotonic to RBC. 0.9% NaCl solution is isotonic to plasma. Dialysis. Special mechanism employed artificially in removal of solutes whose accumulation or increasing concentration could be potentially deleterious to the body. Occurs when there are two or more solutes on one side of a membrane and the membrane is permeable to only certain solutes. If a concentration difference exists for a solute, and the membrane is permeable to it, then that solute will diffuse through the membrane, while the impermeable ones are retained. A principle employed in the artificial kidney. Measurement of body fluid volume. Total body water can be determined directly by drying the whole animal and measuring the loss in weight. Blood volume has been estimated directly but inaccurately by measuring the volume of blood which can be bled from the circulatory system. However, these methods are inconvenient and necessitate the death of the animal. The general method used to estimate these and other fluid volumes is the dilution technique. A compound is injected which becomes distributed throughout the fluid volume in question. Note, the compound which is injected must therefore be restricted to that particular fluid volume. After distribution of the compound is complete, a sample of the fluid is obtained and the concentration of the compound is determined. The volume of that fluid compartment then given as volume of fluid compartment equals the weight of the compound injected/concentration of the compound after distribution. Unfortunately, as the compound is being distributed throughout the fluid compartment, it may be excreted or metabolized and corrections must be made for this loss during the distribution time. Furthermore, it is difficult to find suitable substances, which are distributed only in the fluid volume under study. The best compounds for the measurement of total body water are heavy water, deuterium oxide, and radioactive labeled water, tritium oxide. Isotopically labeled water distributes itself in a fashion identical to the normal water of the body. Other compounds used include antipyrin, sulfanilamide, thiourea, and urea. Estimation of extracellular fluid volume requires a substance which does not enter cells. Inulin, a large dextrin molecule, is frequently used for this purpose, but sucrose, sulfate, and thiosinate are also used. Intracellular fluid volume cannot be measured directly but must be measured as the difference between total and extracellular fluid volume. Plasma volume can be measured by injecting a substance such as Evans' blue dye, T1824, which is adsorbed to plasma proteins. An alternative way is to measure blood volume with erythrocyte labeled with a radioactive phosphorus, superscript 3 superscript 2p. Plasma volume can easily be calculated from blood volume if the volume of erythrocyte is also determined. The measurement of the various body fluid compartments is not precise. Repetition of measurements when different substances are used often gives variations of several percent in the same animal. Hypertonic solution injected into the same region of the hypothalamus caused the animal to drink, suggesting that certain cells in the hypothalamus or nearby in the cerebral ventricles are sensitive to changes in osmoconcentration, and that such osmotic stimuli cause thirst and drinking behavior. When a dehydrated animal drinks, the thirst is temporarily inhibited when an amount approximately equal to the water deficit has been ingested, but prolonged satisfaction is not attained until absorption and dilution of the body fluids removes the osmotic stimulus to the hypothalamic system. There is considerable evidence that thirst and ingestion of water can also be stimulated by a fall in blood pressure, much as this phenomenon also results in antidiuretic hormone release. Evidence for the role of angiotensin as a stimulant of thirst by direct action on the hypothalamus provides a second mechanism for stimulation of thirst by hypovolemia. This control mechanism also constitutes part of the regulatory system for extracellular fluid volume, since fall in extracellular fluid volume will generally result in fall in blood pressure. Fluid and electrolyte balance. The total amount of water and electrolyte composition in the body remains relatively constant from day to day, reflecting balance between intake and output. Intake. Ingested food. Ingestion of water. Metabolic water. Output. Obligatory loss, cannot be controlled. Respiration. Perspiration. Insensible, transpired. Sensible, sweat. Fecal and mouth evaporation. Milk secretion. Facultative loss, controllable, usually through the kidneys.